Okay, so this is going to be my sort of run through of note 9. A lot of you will likely have blanks, so you're going to want to fill out my definitions. My definitions will be put in green, but if you have the textbook, which you can get at the campus shop, if you look at 10.2, everything is just out of 10.2 as well. There's also the 3D anatomy sort of tour, and you can also watch any of the following videos. They're all posted on Edsby, so even if you can't click the link, you can use the Edsby link. If you remember back to last class where you all measured out your digestive system, you should have your chart filled out. So if I scroll down, this chart should be filled out. And the goal I wanted you to see here is think about how long your string is. I think majority of you were around 8 to 9 meters. That means your digestive system, 8 to 9 meters of tubing, fits between your mouth and within your core, right? So that point, this one tube that goes throughout your entire body, if you were to stretch it out, that's a long sort of length, right? And the reason why our digestive system is so long is going back to original things that we talked about. So surface area versus volume. Your body's trying to maximize the amount of surface area so that there is more space for nutrients to get absorbed. Your body's trying to optimize the amount of nutrients it takes in. The reason that's important is because that's the point of the digestive system, to break down larger molecules into smaller molecules. So let's start at the top with the mouth. Okay, you are responsible for knowing these 3 glands. Okay, you need to know where the saliva comes from, right? And this is all the saliva that goes into your mouth. So your saliva consists mostly of amylase, which if you remember back, helps break down carbohydrates. So when we did the Benedict's reaction, we were looking at carbohydrate breakdown. And so that is what we start in digestion, we start with the mouth. So there's mechanical digestion with your teeth and chemical digestion by the amylase in your mouth. And the amylase is from your saliva, which all gets released in different ducts. So at this point, I hope you've written down the 3 definitions. If not, pause the video, write them down. There's value in writing out these definitions because it will help you learn better rather than copying and pasting it, because you actually have to read through the entire sentence. So once the food goes through your mouth, it then goes down. It's been chewed up, mixed with saliva, it's been ground up by your teeth, and then it goes through your esophagus. Okay? And your esophagus is a muscular tube which food passes through. And think about how it moves food down. For one, there's gravity. So gravity helps it out, but there's also muscle, and these muscles contract in a wave-like fashion. So if I were to say, if you held your hand in a fist and then your other hand in the fist and you kept moving one in front of the other, okay, so this wave-like motion is pushing the food down, and that's called peristalsis, okay, sorry. And with that is how you get your food down your esophagus. It's pushing one at a time. Think about if you had something stuck in a very long sock and you just kept pushing it through the sock, that is basically the motion your esophagus goes through to get it to the next part, which is your stomach. I'm sure you've all heard different things about the stomach, but at the end of the day, it's sort of like a J-shaped organ, and this is where food temporarily is, and both chemical and mechanical digestion take place here. So you have, from a mechanical standpoint, you have muscle in there. This muscle contracts and relaxes. By having the 2 contract and relax, it's kind of like it's stirring your food around. So it's what we would call churning in the scientific terms, and that's mechanically breaking down the food just by that stirring motion. But there's also a lot of enzymes that come in to the stomach. The main one you'll hear a lot about is HCl. That's hydrochloric acid. That's a big one in maintaining your stomach pH. pH is a concept you'll talk a lot about in chemistry. And the stomach also has an enzyme called pepsin, which is important for proteins, right? So pepsin will break down proteins into their polypeptides, which if you remember from the macromolecules unit, once we break it down into the polypeptides, eventually if you really want to break proteins down into their amino acid subunits, then you're going to eventually have, it'll break it down into the chains. Once we're in our chains, then you'll break it down into our individual amino acids. And that's the goal of digestion. So stomach kind of kickstarts it. You're just hydrolyzing proteins into the polypeptide chain. So we haven't actually hit the amino acid stage yet. So they're not in their monomers. Then you have our gastric juices. That's anything sort of liquid, enzymes, water, all of that, and that's to help with the digestion in the stomach. It also helps create something called chyme. Chyme is the liquid that anything that leaves the stomach from this point, that is chyme. So it's a thick liquid because you've kind of stirred it all around in this liquid. Anything that leaves the stomach from this point, it's no longer food, it's no longer your cookie or whatever you ate. It is now chyme because that is the makeup of all the enzymes and the digested food all mixed together, and I'm sure you have a lovely image in your head about what that is. Now, the pyloric sphincters, similar to valves in the heart, sphincters are things that help regulate the flow, right? So when closed, this keeps food in the stomach. So you can imagine in a churning motion that if you keep the food in the stomach, you're not losing it. You're able to really break it down and stir it up. The minute your valves open, well, then suddenly you have some loss of food to your small intestine, things like that. So the valve closing is actually pretty important to maximize your digestion in the stomach. Then, once you move on from there, once the valves are open, we move on to the small intestine. There are three main parts to the small intestine. Small intestine is where most absorption occurs and most breakdown occurs. It's pretty important. It's also why, if you remember back to our activity, it was the longest organ. It was 4 times your height. Why is it so long? We're maximizing surface area. We're maximizing the space that your body can digest the materials. So in your small intestine, you have a lot of different things going on. There will be enzymes, you have good bacteria, all of this stuff helps with digestion and absorption. So with your small intestine, that's where your food heads next. And there's 3 segments of it. You have the duodenum, right? And you also have the jejunum and the lumenum. All of these either digest or absorb nutrients depending on the location. The way I've listed it out right now is in chronological order that the chyme would meet each section of the small intestine. Okay, so this is the chronological order that the food would encounter parts of the small intestine. At the same time, in the small intestine, you also have pancreas and the gallbladder, which are sort of accessory organs that help out and produce enzymes that get used in the small intestine. So that is why I have reference to it on these definitions here. But a lot of this is breaking down and absorbing, breaking down and absorbing. So when you look at your small intestine, you'll also notice something called villi. Villi, once again, help maximize surface area. They're like finger-like projections that go around the small intestine. If you look carefully, the small intestine has a sort of curve shape, like this wave-like structure here, right? That curving, once again, increases surface area. We are increasing the area in which food can be absorbed. By having these finger-like projections, so these parts right here, these finger-like projections also maximize surface area, because not only do you have these folds here, but now you have little folds within there that increase more surface area that can absorb nutrients. On top of that, on your villi, if you look very carefully, there's something called microvilli. These are finger-like projections that exist on the villi. And this is when, if I were to look at the cell individually. So generally with a microscope, it's hard to see the actual cell, but generally you'll be able to see the villi very well. But to see microvilli, you've got to really zoom in. And that is looking at, if it's to scale, if you think about it, this area right here is a small square here. So it is very, very zoomed in. Okay, I'm going to leave you guys to answer that question on your own because I think you should know, hint, hint, looking at this and replaying the explanation I just said. Okay, then we have what we call the accessory organs. So the small intestine, there's still more to our digestive system, but the reason why I've put in the accessory organs here is because they work together with the small intestine. So you have your pancreas, your liver, and your gallbladder. A lot of the time, these three pieces are the main site to produce enzymes and send that over to the small intestine. Okay? So the pancreas has something called pancreatic fluid. That has a lot of enzymes that digest carbohydrates, lipids, and proteins. Yes, you need to know that. The liver is the organ with a lot of enzymes. And if you think back to the demo, when I put liver in hydrogen peroxide, it broke down the hydrogen peroxide very quickly. Once again, because it has enzymes. But it also secretes something interesting called bile. Bile is important because it helps break down fats. Digestion of fat is quite difficult because of, for those of you who've taken chemistry, it's because of its non-polarity. It's a non-polar molecule. So the details of that you won't have to know until after you learn chemistry. But bile is needed to help break down fat because we can't just break it down on our own. It has to do with the way it bonds. It's a little more complicated, which is also why I'm sure you've all realized fat is one of the hardest things to lose. So when you eat, you have to go through carbohydrates, proteins, all of that before your body will start breaking down fat. Now, you have your gallbladder that stores bile in between meals. So say you don't use it all, right? Then you have to store some for later. So here's a small explanation in simplistic words of how bile helps to digest fat. Because fat is insoluble, so it is not soluble in our blood or in the liquid in water. So if any of you know how detergents work, bile works by surrounding the fat, right? So it physically breaks the fat droplets into smaller pieces, and then they can get dispersed in the chyme. But what is more important here is if you think about it, if we have a fat molecule here, so that's our fat, the bile sort of surrounds it, right? And this makes it more soluble in our liquid. And by doing that, it breaks it down into smaller pieces, exposing its surface area. Again, surface area is a key. If we expose more surface area, it's easier for the enzymes to find the site they need to break it down. And for those of you who have forgotten, it would be a lipase that would break down the fat. Okay, and then moving on to our large intestine. This is now, a lot of you have asked, why is it called the large intestine if the small intestine is the longest? And that's because the large intestine is wider. So its diameter is much bigger than the smaller intestine. And here, its main job is to absorb the water. There's a lot of bacteria in here, so sometimes if there's undigested sort of plant-based things like cellulose, you have some bacteria that can help out, and it'll help digest things a little further. It also basically takes whatever water was not used up. That water, we're trying to absorb it back because our body is very dependent on water. And then we go to the rectum, which is temporary storage for feces. And this is the final stage before your feces will exit your body through the anus. And that is your large intestine. You don't need to memorize this anatomy. Okay, this is more for interest and also for you just to see the shape. But moving on, you will have to know this diagram for your test. This is the anatomy you are required to know. There's some crossover with the pharynx, from the respiratory system, but you do have to know all of this for your test and for your exam. This will be the diagram used. Notice that I have taken out the sphincters because they're hard to see. It'll also be valuable to know this diagram before we do our dissection.